My name is Alexander Tianara. I'm from Indonesia, and I recently completed my MSc program at Royal Botanic Garden, Edinburgh. And today, I'd like to share my work on revising the ginger genus called Epiamomum. So this genus is supposed to be endemic to Borneo. It's very fascinating for its diversity in bracts and flowers, as you can see from the photos, and its distinct dorsiventrally compressed fruit. So the genus was originally established from the attempt to resolve the taxonomy of Amomum sensulato, or as cardamom. And then the authors, Poulsen and Škorničk, decided to create a genus based on a clade that only consisted of two species, one terrestrial but stilt-rooted and one epiphytic, which inspired the genus name. They later accepted other four species based on the description. And the genus is said to be endemic to Borneo, but the most recent record from GBIF, which is today, showed records from Sumatra, which challenged the previous understanding, and the members are generally threatened, with two are least concerned. So with these objectives and outputs, my study aimed to revise Epiamomum by examining morphology, conducting the phylogenetic analysis, building the taxonomic account, and then updating the species distribution and reassessing the conservation status. I examined 129 collections from 12 herbaria, totaling 208 specimens. And one of the major challenges in my study is the fact that I mostly had to deal with printed specimens, which made up like 77% of the total specimens. From those collections, 101 are Epiamomum collection, are Epiamomum, and 12 of them were used as the material for phylogenetic analysis. So using the ITS datasets, I confirmed that Epiamomum is truly monophyletic genus with divergence during the around Miocene time using the simple data phylogeny program. And the phylogeny revealed two distinct subclades, which is terrestrial and epiphytic, which is very interesting, separated by stilt roots and a unique sheath, which is a cross-ribbing feature. And then the reciprocal illumination helped link molecular and morphological data, which refines the species identification. And as referred in the beginning, species of Epiamomum were the new combinations from Amomum species in 2018, and my work validated 6 species, including one new synonym and one novel combination. I really like to share like every case of the taxonomic problem for each species, but due to the time limit, I'll only share 3 of them. So it was quite naive of me to think that I had to only like deal with 20 Edinburgh specimens, because at the end the number multiplied by tenfold. So I developed a sorting system, a makeshift sorting system, which eases me to detect duplicates and then maybe like repetition in my informal groups and so on. So this is the preview of the refined sorting board. And for species epiphyticum, specimens was very variable, but I managed to detect 4 main forms of spike shape, which seemingly to be different species. And then we can move them to these blobs, I would say. And then I further found 3 special collections that contain 2 different forms on one sheet, which then connects these form blobs. And from these blobs, we can easily detect the duplicates from different forms, as shown in these color pairings. So these are one species, and the shape is more likely due to age. And then this is the synonymic case. It's generally because of the measurement error. So generally, species hansenii is morphologically similar but smaller than pungens. But it's distinctly, the one of the major difference between two species is the length of the labellum, which is supposed to be 1.5 centimeter versus 0.8 centimeter. But we examine the holotype for the pungens, which is housed in Edinburgh, and it never shows any measurement result that near 2.8 centimeter. It's either 0.5 centimeter or 1 centimeter. So basically, the concept of hansenii and pungens is very overlapping, and then to make it as like a strong synonym, I need to access hansenii holotype in Copenhagen to confirm which is if the decision is correct or not. And then the third case, which is the new combination. So this new combination was detected unsuspectedly. The specimens of this species was almost put as undescribed, and it resembles young epiphyticum like I discussed before, but definitely different by having acute brag. And for my final attempt, I tried to read through all the Bornean Epiamomum description, and I came across this species. It was placed in Incertae studies, including the 2018 study, and my study unexpectedly incorporated the specimens into the phylogenetic tree and confirmed it as Epiamomum. Although fresh material and samples are obviously needed to confirm. And from my study, I mapped around 94 occurrence points, adding 65 new ones. The updated map highlights species concentration in northern Borneo, which is Sarawak in Malaysia, but stark collection gaps in Indonesian regions, which is Kalimantan. Conservation assessment and of all species are threatened, reveal all species are threatened, largely due to logging and agriculture if we compare to these maps. And then how about those exciting Sumatran records? And apparently one was a georeferencing error. So it's supposed to be located in West Kalimantan, but was put in Riau province in Sumatra. And then we are left with these interesting records from Leuser National Park in Aceh province. And these are the wilde collections, I usually call them, from the collectors. And Jane Group, who worked on Sumatran Amomum, put the ID as Amomum affine roseisquamosum, because it is indeed resemble that species, with the major difference as being terrestrial, which the real Epiamomum roseisquamosum is aphid. And then who are they? So I found interesting records from similar locality in iNaturalist that resembles those specimens. And in that area, two species of Amomum with stilt roots were recorded, namely Centrocephalum and Apiculatum, which are the closest relative to Epiamomum or the genus. And then excitingly, I managed to find a novel micromorphological character that is likely to be diagnostic and help clarifying the identity, at least for the purpose of my study. So as you can see, cross venation were found in both collections in De Wilde Dives 18059 and 12763 from Q. And then I compared the venation from Centrocephalum and Apiculatum from Edinburgh collection, and I compare it to the Epiamomum specimens. And then from this study, I can confidently emphasize these 3 points: that the Wilde collections are not Epiamomum, And then the collections were identified as Epiamomum affine centrocephalum for now. And then Epiamomum is still endemic to Borneo. So the conclusion for my study is the first one, Epiamomum is truly a monophyletic genus divided into two subclades. One is epiphytic and one is terrestrial. And then the second would be the study accepts at least 6 species and probably like 2 new species in the future if we get the fresh specimens, and then maybe 1 new genus. And then the third is Epiamomum is an endemic genus to Borneo. And then the last is all species are threatened due to logging and agriculture. This study advances understanding of Epiamomum with implications for taxonomy, conservation, and evolution of epiphytism, especially in Zingiberaceae, which is not a common phenomenon. It also underscores the importance of plant taxonomy in supporting conservation efforts in Borneo, including Malaysia, Indonesia, and Brunei Darussalam. So I would like to thank my sponsor from the Indonesian government and to everyone from these herbaria in these countries, and then to these kind of people that helped me to go through the MSc project. And then I would like to thank the support from the Civil Trust project number 2024 #24. And then I would like to dedicate my work to the authors of the genus and species, Mr. Schumann, and then Rosemary Smith, Sakai, Nagamatsu, Axel Poulsen, and Yana Leong Škorničkova. And I also would like to dedicate my work to the past collectors, including the unrecorded indigenous people of Borneo, who have made the collections and taxonomics. Ya, terima kasih. Thank you.